Okay, so for number one, it says that an individual is heterozygous for the following inversion, and so it gives you the normal chromosome and then the inverted chromosome. And so we just, I put this normal one up and duplicated it because it says that the two chromosomes replicate prior to meiosis. And then we know that the cross is happening between the S and the T allele. And so for the inversion chromosome, I put S and T, lined it up, and then this was the inverted chromosome. So everything before S, I wrote that which comes before it, and then everything that comes before T, and then I duplicated it. And so we have the two normal ones for our first two products, and then the cross, I just went through these letters, and then where S, you move down to the lowercase t, the inverted chromosome, and then you go same way this way. And so you'll end up with one that doesn't have any centromeres and then one that has two centromeres. And so those are your 4 final products.
Alright, so for question 2, we're doing the fill in the blanks. So part A says non-disjunction in what produces 50% aneuploid gametes. What do we put meiosis 2 because it'll have half of them will be normal and half are trisomic and monosomic. And it says whereas non-disjunction in mitosis can produce a mosaic phenotype because they split into monosomic and trisomic and then continue to reproduce as monosomic and trisomic, so you have kind of a mixture of both of those. And for part B, we have meiosis in an individual who has suffered a paracentric inversion, produces a dicentric chromosome and an acentric chromosome because it causes duplications and deletions, making one have no centromeres and one has 2 centromeres. And then for C, we said Turner syndrome is an example of a human disease caused by monosomy because there's only one X for the girl in chromosome 21. And familial Down syndrome is an example of a disease caused by Robertsonian translocation because there are 3 long arms of the chromosome 21 causing Down syndrome. And for D, we said in human tissue, specifically in the reading it said the liver cells can be 4N, 8N, or 16N. And then E, a terminal deletion in chromosome 5 causes Cri-du-Chat syndrome, which is just the arm of the chromosome is deleted, while an inversion on the X chromosome causes type A hemophilia because the blood clotting gene breaks during the inversion. For F, duplication or gene redundancy is a change in chromosome structure that can be potentially beneficial to an organism or an entire species because it can help produce more things than having just one. And for G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offspring because the translocations are balanced. Alright, first question number 3. For part A, it says how many total chromosomes would be found in the humans that have the following conditions. And the first one is Turner syndrome, which will have 45 chromosomes because one of them is deleted. Jacob syndrome will have 47 because there's an extra one. Cri-du-Chat syndrome has 45 and a half because part of the chromosome is deleted. Familial Down syndrome has 47 because it has an extra. And type A hemophilia has 46 because the inversion doesn't change the number of chromosomes. And for part B, it asks which of the following terms is not associated with Charcot-Marie-Tooth disease type 1A. And we said triploid, reciprocal translocation, and diploid are not associated with that. And then for part C, it says which of the following terms are associated with Klinefelter syndrome. We said aneuploid because there's a change in chromosomes, trisomy because there's an extra of one of them, non-disjunction because it happens during meiosis, and duplication because there's an extra copy of the chromosome. And then for part D, it says which of the following terms are associated with type A hemophilia, and we said non-disjunction because the change happens during meiosis.
Okay, so for question number 4, we're going to look at a translocation segregation. And so what we did up here is we just simplified it, so we only show one strand. We don't show the sister chromatin attached, just because it made it a little simpler. And so for part A, we're looking at the diagram right up here, and here's each individual chromosome and how it's going to line up for the segregation. And then to show question B, we're looking at the alternate segregation. And so what we're going to do is we're going to take the chromosomes that are normal, and those are going to be with each other. So you can have these 2 chromosomes together in a cell, and then you can have these chromosomes in a cell, and that produces the alternate segregation. Then if we're going to look at the gametes, they would divide one more time, and we'd end up with each one of these as an individual gamete. Again, we just kind of simplified that. And then for adjacent 1, we're going to look at the cells that are both next to each other on the bottom and on the top, and those are going to get grouped into a cell. And so for the top one, we have these chromosomes and these chromosomes, and then the bottom, these ones and these ones. So again, the gametes would end up being each would be one of these, one of these, one of these, and one of these. And then adjacent 2, very similar to adjacent 1, but we're going to take these chromosomes next to each other and these chromosomes next to each other as well. And that's what gave us our products here for adjacent 2, and that shows the gametes there and how they differ. Alright, so for question number 5, part A, we're trying to find the 3N, or the triploid number for the cat. We know 2N is 38, so we're going to divide that by 2, N equals 19, multiply by that by 3, and our triploid number is 57. For part B, we're looking to see what happens for a cat that is monosomic for chromosome 12. So that means it's only going to have one copy of chromosome 12, so it's going to be one shorter than what it normally is at 38. For part C, we're looking at a cat that suffered a terminal deletion on one of the chromosomes. And so that's why we said it's 38, but we put a little asterisk by it saying that one of the chromosomes is going to be shorter because there was some deletion taking place in that chromosome. But there's still a total of 38 total chromosomes. Part D, it says the cat suffered a Robertsonian translocation, and that's going to be 38 still because the chromosomes are just switching some pieces together, but the total number is still there. And then for part E, a reciprocal translocation between 2 chromosomes, same thing. We're going to have some switching around, but there's still 38 total chromosomes.